Hey everyone, today I'm going to tell you all about aquaporins. Imagine your body as a bustling city with a network of roads and highways connecting to different neighborhoods. Now think of aquaporins as the high-speed lanes specifically designed to transport water molecules in and out of your cells. In this video, we're going to explore the world of aquaporins as the expressways of cellular hydration, ensuring a smooth flow of water traffic for the well-being of your body's infrastructure. Let's dive in. Aquaporins are the essential proteins in our cells that act as the molecular water channels. Their primary role is to regulate the flow of water in and out of our cells, ensuring optimal hydration levels. This selective permeability is crucial for maintaining the health, the function of cells, and they play a vital role in preventing dehydration, supporting organ function, and contributing to temperature regulation within the body. So basically, they're like the efficient transportation system that keeps the cellular environment well in balance. The goal of my project was to conduct a comparative study of the structure and sequence of 4 aquaporins from different species: human, bovine, E. coli, and E. coli glycerol facilitator. All of this was using the multiple sequence alignment tool, or multisec, and VMD, but we'll get more into this later. This project was meaningful in 3 ways. 1. By looking at cells in various animals, we can learn how they changed over time, giving us clues about how they've evolved, and we'll actually be able to see that later. Secondly, aquaporins move water around in tissues, and comparing the 4 reveals similarities and differences. And finally, using high-tech tools like multisec and VMD shows how this kind of technology can help us understand complicated biological structures in the future. So now let's take a look at the lab. And I followed the tutorial that I found from the University of Illinois training. They have many detailed tutorials that scientists can use to work on their own pace, and many of them will use the VMD lab. For example, the tutorial that I did was 84 pages, so they're definitely really detailed. And as you work through these, you'll get a solid understanding of the lab. First, I downloaded all of the necessary software, so the VMD lab, the multisec analyzer, and the user's guide. I loaded all of the molecules in that we were working with, and you can see that here. I then started modifying the representations and the residues and examining how that impacted the view of the protein. By changing these representations, I could see the different proteins within the aquaporin. When examining the alignment, I opened the multisec window tool, which looked like this. I loaded all of the proteins in once again and aligned the molecules by clicking on Tools, Stamp Structural Alignment. See how when I align them, they look different and they look more together? That's because they are. Alignment is a measure of order in which molecules are arranged or placed with respect to axes fixed in space. Multisec also provides tools that allows you to color the molecules according to their Q value, or QRes, a measure of structural conservation. Conservation indicates that a sequence has been maintained by natural selection, and a multiple sequence alignment, which is multisec, for us is the 4 different species. You can see that the residues that are conserved across all sequences are highlighted in different colors. Take a look. I could also sort the different molecules based on which ones had a high value of QRes and which ones didn't. In this step, I created a graph of RMSD, which shows per residue values, and compares 2 proteins to see how well they align. It's pretty cool. Finally, I loaded in one of my molecules and examined the tetramer, which is a molecule that consists of 4 structural subunits. So basically, when you use the tetramer, you could see the 4 structural subunits of your molecule. However, this one was a bit harder to remake and show you guys. And that's it. Aquaporins in this project have the potential to strongly benefit industries, but one specific one is the pharmaceutical industry, by providing insights into the structure and function of aquaporins. Understanding the variations across species could lead to the development of targeted therapies for conditions related to water balance and cellular hydration. Additionally, the use of advanced computational tools sets a precedent for the integration of technology in the biological research, potentially advancing innovation within the biotech sector. This holds the promise of transformative breakthroughs in medicine and technology, marking a crucial milestone in our relentless pursuit of understanding life at its fundamental level.